Hello, and thanks for joining us today for this Cosmo Bio USA webinar. My name is Matías Jauregui, and I will be your moderator today. In this webinar, Dr. Jasmine Hershewe will present about how to get to the right clone faster, new approaches to complex Gibson assembly. During the presentation, if you have any questions, you can submit them using the Q&A button at the bottom of your screen. Please put your questions here and not in the chat window, so we can be sure to answer the questions during the Q&A session following the presentation. Also, we are recording this webinar, so if you miss any part of it or you want to rewatch it or share it with your colleagues, that will be possible, and you will be notified by email when the video recording is available. So again, thank you very much for joining for today's webinar. And without further ado, I will pass to Dr. Jasmine.
Well, thank you so much for the introduction, and thank you to everyone who's joined today. I'm very excited to be here from Racer Biosciences to talk to you a little bit about getting to the right clone faster and some new approaches and reagents for complex Gibson assembly. All right, so I think we can all agree that we're living in a very exciting time where the pace of biological design is accelerating very rapidly. And this is in part due to the advent of AI and machine learning tools that are enabling us to generate more and more high quality hypotheses faster than ever previously possible. And this is really leading to a paradigm shift at the bench where we're moving from techniques like changing one thing at a time, locking and move on, and more towards high volume, high precision, and high parameter testing. So instead of random methods, maybe where we're kind of throwing darts at a dartboard, like you see on the left here, we're developing approaches to really quickly and efficiently move through these complex design spaces that are indicative of biology. But with these new design workflows come a lot of new challenges for the actual building and the testing of the molecules. And a very critical piece of that workflow to focus on is the DNA building process. So AI gives us this massive universe of things to test, but almost immediately the reality sets in, our ambitions are squeezed, and our budget, our throughput, and our physical constraints sort of funnel all these exciting things that we can test. And because DNA is the starting point for most biological systems, it's kind of the first choke point in this funnel. So opening up this funnel really requires us to have robust, high throughput, and modular workflows for DNA building. And that is the topic of today's discussion. So today, just a quick roadmap for the talk. We'll be going through multi-fragment Gibson assembly as a design strategy, with the core idea being that with reliable multi-fragment workflows, we're able to open up new opportunities and cost savings. I'll introduce you to our reagent called F1-X, which is Next-Gen Gibson Assembly, that allows us to very efficiently clone many fragments in a single tube, like you're seeing in the bar chart here, with cloning efficiency over 95%, even up to 11 fragments at a time. I'll go through a couple of case studies, and then we'll close it off on practical tips for getting started with F1-X. All right, so just to set the stage, just taking a 10,000 foot view, something that we hear all the time, a question in the field is, why should I stop outsourcing my cloning, and should I even be cloning at all, which are totally fair questions. I think many people sort of gave up the idea of doing cloning or high throughput cloning in-house years ago because of complications with the DNA ecosystem. So maybe you experience things like weeks or even months long lead times for fragments, and really not a lot of options for obtaining complex DNA sequences to your doorstep for DIY cloning. The cloning chemistry itself was often limited and could be, not very robust and fail. And then on the sequencing side, high cost, long lead times, needing to design primers and do mini preps for every single Sanger sequencing reaction. And if we fast forward now to 2026, a lot of this has changed, and there's a very active ecosystem of vendors around DNA that are doing a lot of great work to speed this stuff up. So you can now get custom fragments on your doorstep in days, not weeks, and there are plenty of vendors now that are able to make very complex DNA sequences that enable you to do cloning in-house with high complexity DNA. The cloning chemistry itself, the reagents, can now really effortlessly handle what used to be pretty high risk or a pretty big chore in the lab. And the advent of next-day primer-free whole plasmid sequencing directly from a colony on a plate is now available. So that's how all of the data in this presentation was generated, and it's really changed the game for me and for many people who clone regularly. And so all that to say that building in-house is now a much more realistic option. It's a much more tractable option for saving costs and enhancing the throughput of your builds. And so we think that this may call for a little bit of a mindset shift around cloning. So from cloning being sort of a rate limiting lab chore to thinking of DNA assembly as a tool that can really power your science. And we'll be talking about some aspects of this today. So the topic of today's talk is Gibson Assembly, which is a simple and a seamless method for joining together DNA fragments. And you can execute Gibson assembly in these 3 simple steps. So first, you prepare your DNA, linear double-stranded DNA fragments with designed overlaps that we'll talk more about later. In step 2, you mix those fragments together, and in the single tube, you add your reagent. Let that incubate between 50 and 60 minutes in a single tube, single step. And then you are ready for your downstream, whether that is a transformation or a cell-free workflow. And to zoom in a little bit more on what's happening inside the tube and talk briefly about the biochemistry, I just want to talk a bit about how Gibson assembly works. It has quite an elegant mechanism. This was invented in 2008 by Dr. Dan Gibson. And in this tube, we really have 3 core enzymes that are doing their functions in concert all at once. So if you take a look at the schematic here, we're going to have a very simple example of 2 fragments with designed overlaps that are shown here in pink and in green. So when we mix these fragments together with the reagent, there will be an exonuclease that will chew back one strand of the DNA, which kind of exposes these overlap regions as single-stranded DNA, which is now sort of like a custom sticky end that is free for base pairing across the strands. Those chewed back overlaps then anneal together, which forms a primer sequence for a polymerase to come in and fill the gaps, and then a ligase seals the deal. So at the end of this reaction, you'll have 2 fragments that have come together in a directional reaction. But this is actually useful for many more than 2 fragments at a time. This is just a simple example. And because of that very simple, seamless chemistry, Gibson assembly has many advantages over different cloning techniques that are out there. So here I'm showing you a spectrum from simple, maybe 1 fragment or 2 fragment assemblies where you don't really care about the direction of the DNA that's going in, all the way up to very complex assemblies. And Gibson assembly is very versatile. So it's good for both very simple and moderate to complex cloning tasks, the same exact reagent. It's a simple system. So with some different technologies, like for example, restriction cloning, you actually have to choose enzymes off the shelf from a catalog of reagents. You just have to make choices, which can lead to some extra work and even some design fatigue. There are no complicated inputs or multiple steps that are required to do Gibson assembly. And most importantly, I think, and the big innovation of Gibson assembly when it was introduced, is that it does not introduce any scars or mutations through the assembly process. So the assembly process is inherently seamless or scarless, and that's what we mean when we say that. And Gibson assembly in recent years is gaining a lot of momentum with researchers who focus on high throughput and AI. So here's an analysis that we did of articles in PubMed Central where we're looking for keywords of Gibson assembly in the methods section. Year over year, we're looking at papers published. And what we can see is a sort of hockey stick shaped graph where there are more and more publications year over year. And then we cross-checked for concurrent use with terms around AI and high throughput screening, which are shown here in purple and yellow. And what we can see is that the curves are very similarly shaped. And just as a time point here, in 2023 is really when ChatGPT sort of launched in earnest, which is sort of the pickup of generative AI tools. So we're definitely starting to see trends across many fields: antibody discovery, enzyme engineering, cell and gene therapy, metabolic engineering, basic biology, where Gibson assembly is being used more and more in-house to accelerate the builds and to move into more high throughput work. And despite its vast usefulness, these first generation Gibson workflows, again, while being incredibly useful, do have some bottlenecks. So one thing we hear in the field all the time is that users start to feel constrained around 3 or 4 fragments assembling at a time. And so what we really need is higher efficiency and predictability with multi-fragment workflows, to keep an ultra high fidelity with more and more fragments, and to keep the simple one-step workflow with more and more fragments. Ultimately, taking the same workflow that we know and love and just pushing the limits a little bit of the complexity of reaction that we can run with a single-step Gibson assembly. So this is why we created F1-X, which is our next-gen Gibson assembly kit shown in the picture here. The kit comes with a master mix, a positive control, and a quick start guide. It comes in a variety of sizes here, including bulk format. So this is the same core chemistry I talked about earlier. It's known and loved and cited over 40,000 times. And it's engineered with headroom in the specifications. And what I mean by that is we push the limits of things like scaling down the reaction so that you can get more out of a single tube of master mix. We offer a wide range of fragment sizes that are able to be put into the reaction, between 100 base pairs all the way up to 32 kb, building up to 100 kb of total DNA. You can add robustly between 2 and 12 fragments in a single one-step reaction. We've even stress tested all the way up to 14 successfully. We've also noticed there's a pretty high tolerance for higher temperature in this reagent, which can be useful, especially if you're trying to assemble complex DNA. Maybe you need to melt hairpins or do a little bit of a extra finessing to get the assembly over the finish line. And it's compatible with different additives and different downstream workflows. So again, I don't mean to rattle off specs here, but the idea is that we've engineered this reagent with headroom to give you freedom to develop workflows that help accelerate your science. And I do want to note that this reagent was developed with the inventor, Dr. Dan Gibson, who is an advisor to the company. So now let's dive in a little bit on use cases that can benefit from more robust multi-fragment cloning. And I think really any biological system that can be pulled apart and studied at modules or at the module level, whether it's in the construct, so things that are not coding sequences like UTRs, regulators, ribosome binding sites, or the cargo itself, so maybe different domains of a specific protein that you're studying or that you're interested in. The ability to do multi-fragment assembly really lets you think combinatorial, and again, in modules, so that you can decrease your synthesis cost while testing more and more designs. And we do see common use cases all the time around drug discovery, especially with the advent of nucleic acid therapeutics that really benefit from cis elements in the sequence, metabolic systems, metabolic engineering, and basic research. So antibody discovery has historically taken advantage of sort of a modular cloning approach for many years to optimize their discovery economics. So here I'm showing you a schematic of an antibody where these regions here, sort of at the tips of the heavy chain and the light chain, are the variable regions. And so what's done is, instead of synthesizing the entire chain of the protein, all you need to do is synthesize the part that's changing, which can drastically change the economics of your assembly campaign. So for example, in a campaign with 96 heavy chains and 96 light chains, you can save 67% on your DNA synthesis costs by only changing the part that varies. And while an antibody is a relatively simple example of this, because there's just sort of one module at the terminus of the protein that needs to change, many use cases require more than one variable domain to achieve the right output. So here's an example of a protein that is beloved to me. I worked on this protein in grad school called PGLB, where if, for example, I was interested in changing the substrate specificity of this enzyme, which does glycosylation, I would need to change a lot more than just one piece on the terminus. In fact, I would need to change multiple pieces. So here I'm showing you sort of a zoom in on the crystal structure of the substrate binding pocket, where there are residues around 44, 469, and 435, let's say. that all participate in this binding. So if you stretch that out across the sequence, you can see that you may need to actually change multiple modules at a time. And with tools like AlphaFold, so I'm showing you the AlphaFold prediction here on the left, this type of analysis is becoming more and more common to be able to do at your computer without a crystal structure. So if we think about the ways that we could engineer this protein or unlock some engineering here with a two-piece assembly, we may be confined to doing something like an alanine scan or an NNK scan, where we would have to synthesize up to 1.5 million base pairs of DNA just to screen 711 variants. This protein is 711 residues long. Whereas if we move to a modular design, where let's say we take all three of those modules that we're interested in the substrate binding pocket, and we introduce 500 precision variants at 20 residues per motif, now you're looking at synthesizing orders of magnitude less DNA. So less than 150,000 base pairs of DNA to screen 500 to the third combinations. And so what that gets you is about 6 orders of magnitude more efficiency on your variants that you can make per dollar. And now you can see that the bottleneck really moves to liquid handling and assay ability if you have the ability to clone in a 7 or 8 piece assembly and make this a modular engineering exercise. And so now bringing this graph back that I showed you earlier, I think this puts into context a little bit better why we care about the ability to clone more and more fragments. So here is some performance data from F1-X, where I'm showing you cloning efficiency as a function of the number of fragments that we're cloning at once. So 4 fragments, 7, 9, and 11 fragments. And what we can see is a consistency across this fragment range and a high cloning efficiency. But efficiency is not the whole story when we're cloning. Obviously, we need to make sure that we get plenty of colonies. So even out to this 11 fragment assembly, we can find hundreds of colonies on a plate to screen, depending on how much of the transformation we plate. And then in terms of the error rate, here we like to benchmark against the error rate of the DNA that's going into the reaction. So in this case, simple examples here are these 9 and 11 fragment constructs, which are made completely of synthetic DNA, just straight out of the synthesis plate. We estimate that the error rate is around 1 in 7,500. And when we run this whole plasmid sequencing across a population of clones, we see that the error rates across the populations are actually lower than what we would anticipate the input DNA is. All right, so zooming up a little bit from protein level engineering now to some systems level engineering, these modularity methods are also extensible from protein domains and into full pathways or systems level work. So here's an example of what a construct might look like to test a 3 enzyme pathway, kind of shown in these orange colors here, where we have a substrate that we'd like to convert into a product. And the goal here is going to be to optimize flux through this pathway. So maybe we'll want to change the identity of these enzymes, test a few homologs, which is becoming easier and easier to identify these now with generative AI tools. Maybe we want to change the expression levels by changing these regulators and UTRs. So again, this is just another way of thinking of a construct in modules. And to demonstrate this type of modular engineering, we put together a white paper on the one-shot synthesis of a small lycopene biosynthesis pathway using F1-X. So in these experiments, we took a 9 kb circular construct. In this case, the pathway had 4 enzymes. We split that plasmid up into 14 different pieces with 40 base pair overlaps, with each piece being around 700 base pairs. Here you can see this is the synthesis product that came to us directly from the vendor, where the DNA builds look mostly clean. We have a little bit of low molecular weight contaminant here, but again, pretty clean. These fragments were taken directly out of the synthesis plate and pooled together. You can see that in this lane here. And then when we add F1-X, we see this high molecular weight line is indicative of assembly, disappearance of the substrate, and appearance of higher molecular weight assemblies. But what we were really excited to see was that on the plate, the E. coli colonies, some of them, started turning this orangish brown color, which is indicative of the synthesis of lycopene, which is not a natural molecule that E. coli produces in high quantities. So we know that the pathway is fully assembled, all the regulators are there, the enzymes are there, and it's active. So we took some of these colonies and sent them for sequencing and found a pretty good ratio of full-length product and error-free clones. So all of this really demonstrates, I think, why modular cloning can be a helpful tool. And if you are currently using a cloning product or a workflow that is one inserted, one vector, I would definitely encourage you to think about what you could achieve if you could pull your biosystem apart and sort of probe more pieces simultaneously, and could you save on some DNA synthesis costs in the meantime? All right, so now I want to go through some practical tips for getting the most of your multi-fragment assembly. So here is a traditional cloning workflow where we start with the design. This is usually happening in silico or on our computer. We then need to obtain our DNA fragments. So oftentimes this is synthetic DNA from a vendor or PCR with synthetic primers. We can then QC our DNA like those gels I showed you earlier. There are also quantitative methods to assay for percent of full-length products like a tape station or a bioanalyzer. We then pull those together, run assembly, for example, Gibson assembly. Transformation into E. coli is typically the next step in a traditional cloning workflow, where you now have a plate with colonies to screen. Typically, this is done with sequencing, but you can also do an intermediate step where you screen for, let's say, a full-length insert using a colony PCR. And then some downstream maybe involves scale-up and, let's say, expression of a protein. But I want to highlight 3 pieces of this workflow to kind of go into some details and some tips and tricks for onboarding our cloning reagent F1-X if you're interested to do so. So the first is really around the assembly piece. So choosing a selective master mix or choosing a good master mix. So I would say not all master mixes are created equal for a given construct. So here is some performance data showing F1-X against a few very similar types of seamless assembly master mixes. And what we found was that the average cloning efficiency for F1-X was the highest of all of these different kits. And why that matters is because it helps you get to the right clone faster. So here I'm showing you a curve of F1-X in blue with, in this case, a 78.6% fidelity and a Gen 1 Gibson assembly kit, which gave us about 40% fidelity. And what you can see is that in order to reach 95% confidence that you've picked a colony with the right sequence, you only need to pick 2 colonies with F1-X. However, you might need to pick 6 with a different master mix. And at $15 per reaction, the cost of cloning really is accrued here at the sequencing step, not at the colony counting stage and not so much at the stage of setting up the assemblies themselves. So enhancing your choices of getting the right clone in your sequencing experiments is very important. We also recommend using a robust chemistry that has tolerance and flexibility, especially if you're interested in moving into things like automation. So F1-X has been stress tested against freeze thaws and the addition of crude PCR master mix contaminants, as well as shortened incubation times for many fragments. So here's some data kind of representing that. So in this experiment on the left, we took F1-X and we froze it and thawed it 5 times right in a row. We ran an 11 fragment assembly and we assayed for colony count, cloning efficiency, and fidelity. And what we saw was that there was no drop in performance even after 5 freeze thaws, which gives you more confidence in the reagent as you're using it day to day, pulling it out of the freezer. Don't necessarily have to treat it as if it were not robust. Compatibility with crude PCR mix. This is really important because sometimes you want to save time. Maybe you have a workflow where you're obtaining a small amount of a synthetic fragment from a vendor and you need to amplify up enough to do cloning. If you have a clean, full-length PCR product, you can actually spike that directly into the F1-X reaction. So here's an example of a 2-fragment assembly where we see the unassembled DNA on the left, the assembled DNA on the right. And what we saw from this experiment was that all 10 of the colonies that we picked off the plate were sequence perfect, even with 20% contaminant or crude PCR mix. And then finally, another stress test here where we have a 9-fragment assembly with 20% crude PCR mix, and we've even shortened now the assembly time from 60 minutes to 15 minutes, and we can still see plenty of functional colonies on the plate. Another feature that's very helpful when you're thinking about scaling out and scaling up your build phase is using a chemistry that's miniaturization friendly. So in doing miniaturization, you can actually get much more out of your cloning reactions. So for a standard reaction size of say 20 microliters, where you have a 10 reaction kit, you can run 10 reactions. But if you scale those down to 5 microliters, now you can suddenly do 4 times more. So you're getting 4x more efficiency. Here I'm showing you some data where we're scaling down the volume of the F1-X reaction and we're running sequencing. And what we can see is that the colony count and the fidelity are ultimately very stable, even with multi-fragment assemblies going down to smaller and smaller volumes. All right, so the second piece of that workflow that I showed you earlier that I want to talk about is the colony, and I've crossed out the word colony here purposefully and put the word construct screening. So high level, not every workflow necessarily needs to end with a sequence confirmed plasmid. And I would say that cloning in E. coli definitely has its limitations because E. coli is sort of this very strict biological filter that has lots of conditions that it needs to be successful. And sometimes this filter can create downstream challenges like cellular toxicity. Maybe you just don't get any colonies on your plate because the protein that you're trying to clone a construct for is toxic. Or maybe plasmid instability. We see this all the time with RNA constructs that may have something like a poly-A tail, where the length of the tail will be changed inside of the cell. And then throughput limits. So just by design of needing to transform and plate, this takes a little bit longer than something that can be fully automated all the way through in wells, let's say, on a liquid handler. And F1-X really does enable the use of cell-free workflows that allow you to move outside of the cell. So what I mean by that is instead of going through a traditional cloning workflow, which can take 3 to 5 days between the transformation, the mini prep, and the sequencing, there are other routes to just using your raw assembly product and either putting that into, let's say, a PCR or a rolling circle amplification reaction, which gets you to, let's say, a linear expression construct or a linear DNA in hours. And this can be very useful for going directly into systems like cell-free protein synthesis. So you can go from linear DNA fragments through the cloning, take that cloning reaction directly into a PCR, and take that linear template directly into a cell-free reaction, all in, let's say, an automated fashion. And another great use for linear DNA within this context is the ability to put it directly into an in vitro transcription reaction where the endpoint is then mRNA. All of these workflows, again, taking less time than traditional cloning and being highly automation friendly because they're all well-based transfers. So here's an F1-X workflow that we've tested out, generating a poly-A containing template for IVT. So poly-A tails, as I mentioned earlier, are notoriously unstable in cells. They can be. And this in vitro workflow really kind of bypasses the hassle of needing to go through living cells just for screening, let's say. So in this case, let's say you had a long open reading frame that you wanted to clone. Like, let's think back to COVID vaccine. I think the spike gene was pretty long. So you may need to synthesize that in multiple pieces. You can assemble all those at once using Gibson Assembly. With that crude Gibson Assembly reaction from F1-X, you can then take a small amount of that, add primers. In this case, the primer is going to be the carrier of the poly-A tail. And now you have a linear PCR product of your fully assembled gene with your UTRs and your poly-A tails ready to go for IVT. And for this workflow in particular, linear DNA is in many cases the template of choice for IVT. So you're not really making a trade-off on whether the endpoint is circular or linear. So here's some data around the F1-X assembly. On the left, I'm showing you a gel of the assembly for 1 fragment on the top and a 4-fragment assembly on, 2 and 4 fragment assemblies on the top and bottom. So we do see that Gibson assembly ladder within the F1-X reaction. And then after the PCR, where we're sort of resolving the entire insert, we see exactly what we want to see here, which is a very clean full-length product at 1.7 kb, which is the intended size of this construct. And why this is important to get a clean band here is that it means that the ligation efficiency of the F1-X is high enough. And when you sequence these linear DNAs, what you really want to see is a very low error rate. And F1-X can deliver that high ligation efficiency and the low error rate needed to move forward with an in vitro workflow. And so one thing that we do, discuss all the time with people who are interested in onboarding workflows like these are the trade-offs between a plasmid route and a cell-free route. And while there are trade-offs and maybe there are, let's say, hits in the efficiency at each step, these can all be measured and assessed against the goal. So for example, in the plasmid route here, oftentimes what you'll see is 100% fidelity where almost 100% fidelity, where mostly all of the plasmids in your population are the same sequence. Whereas with the cell-free route, you may have a few sequences that are not exactly the correct sequence, maybe because of errors in the synthesis, but there will be some templates that maybe don't carry 100% correct sequence. So that's a quantifiable trade-off. And then at the level of the protein, whether you're using a cell-free system or transfecting linear DNA directly or transfecting mRNA in place of a plasmid, oftentimes you may see a very predictable drop in the yield of protein or a change in the kinetics of expression, which is not a failure, it's just a change. So these are some of the things that we think about as we're considering onboarding higher throughput workflows with F1-X. All right, and the third and final aspect of that workflow that I want to talk about is designing for success. So the F1-X chemistry was really optimized with a very simple 40 base pair overlap default to keep things very simple. So if you are going to do multi-fragment cloning with F1-X, we always recommend starting with 40 base pair overlaps, which is a little bit different from some other cloning chemistries. F1-X is optimized to efficiently clone over a wide range of GCs on the overlaps. So we've seen success over low GC regions from 30% all the way up to high GC regions of 70%. So here on this plot, I'm showing you for a 2, 4, 7, 9, 11, and 14 fragment construct, 1 point per overlap, where you can see plotted the GC content against the secondary structure. And as the secondary structure gets more negative, this means that in the overlap, there may be a stable hairpin. And even at high GC and with stable hairpins, we do see successful assembly over these junctions, which is one of the things that we specifically engineered F1-X to be able to do. And then one exploratory feature that we're very excited about is that if the assembly, not all assemblies call for this, but if the assembly calls for some troubleshooting, we have a new lever to do that now, which we're calling temp tune. So traditional Gibson assembly is treated as a fixed temperature workflow. Usually you're executing this workflow isothermally at 50 degrees. But with F1-X, we feature a highly temperature stable formulation where you can actually ramp that temperature up if needed to resolve hairpins and promote more productive annealing within a construct. So here I'm showing you some data around the 7 fragment assembly where there's a big hairpin in one of the junctions. And what we see is that the typical 50C reaction parameters is fine. We get about 60% error-free assemblies, which was better than the benchmark that we tested against. However, when we ramped the temperature up to 53 degrees, we saw an increase in the fidelity, and we also saw an increase in the colony count. So this is just another handle to allow you to troubleshoot constructs without needing to necessarily redesign the DNA, just run the thermal gradient and see if you can change the odds. And in the future, one thing that we're really excited about is moving toward more predictive design tools. So instead of using the current methods, which are not necessarily sequence aware, our vision is thermodynamically aware algorithms that are trained directly on real F1-X success and failure data, with the goal being to catch sequence problems in silico before you waste a week at the bench. All right, so to recap, I hope today I showed you that F1-X can enable you to clone multiple fragments robustly. And I hope that you may use it to power your next experiment, figure, paper, group meeting, breakthrough, whatever you have going on. So just a little quick note about Racer Biosciences. We are a small startup in San Diego, and our mission really is to focus on DNA-based reagents that save time and simplify bench work. If you like the things that I talked about today, we have a LinkedIn page where we discuss cloning tips and tricks. We also are running a series right now on where cloning shows up in places in your life that you may not expect. For example, like cheese and glowfish at Petco. So if you're interested in that kind of thing, give us a follow. I want to quickly acknowledge the team that was responsible for this work. And if you're interested to test F1-X for yourself, you can get in touch or go ahead and place the order at cosmobio.usa. Here is the product page for F1-X. You'll also note here in the sidebar on related products, this Gene Frontier Pure for X 2.0, 2.1. These are cell-free protein synthesis kits that allow you to go directly from linear DNA to protein. So if you're interested in piecing together a workflow like this for yourself, we can definitely help you get started. And with that, I'll wrap it up. Thank you so much for your attention and really, really appreciate it.
Thank you very much, Jasmine, for your talk. That was a great presentation. We do have time for some questions, so please remember to use the Q&A feature at the bottom of your screen. So again, thank you very much, Jasmine. And we do have our first question, that is, do you plan to offer a web tool to assess full-length sequences for the best sites to split into fragments? Maybe also taking into account the customer desired max fragment length.
Yes, absolutely. So that's something that we currently have in process that we're really excited about. We're hoping to have a beta launch of that software available in the next coming months. So yeah, go ahead and stay tuned. You can get in touch with us and we can definitely let you know when that launches. But yes, the idea would be to have a tool where not only can you check your work. So if you had an assembly maybe that was a mystical failure, this is something we hear all the time in the field. You can read about it on Reddit too, right? Mysterious cloning failures. The idea would be to use more advanced bioinformatics in order to assess those failures. So, maybe figuring out past failures, but then also finding optimal split points and really making sure that the sites are unique with low secondary structure and optimizing that for the Gibson assembly reagent. Or even telling you when Gibson assembly might not be the right fit for your assembly and you may need to try another technique.
Perfect. Also, another question is, can someone assemble a plasmid without a vector?
Oh, yes. So this is actually, I would say, a big advantage of using a multi-fragment workflow. So typically with cloning, you'll do a vector prep first, which can involve a PCR, deep-in-one digest, a long process, gel extraction, or a restriction digest. But with multi-fragment cloning, you can actually just take your vector and split it up into smaller pieces and synthesize it all in one shot. And this has a lot of advantages because you're not having any background carry through. So usually when you do a vector prep, you have some circular template that can, come through and contaminate the reaction. But the ability to synthesize a whole vector in one shot, I think, is a huge advantage of being able to clone more and more fragments.
Perfect. And also, is there a difference in the protocol between running a simple assembly and a more complex multi-fragment assembly?
Yes, but only slightly. So in general, it's the same high-level workflow where we have a one-step Gibson assembly. We typically define a simple reaction as 2 to 3 fragments, where we recommend an incubation time of around 15 minutes and shorter overlaps, around 20 to 30 base pairs. And then as you get more and more complex, we do recommend a longer incubation time to start and longer overhangs around 40 base pairs. And I think we have a couple of differences in the concentrations and ratios that we recommend that you can find in our user guide.
Perfect. And the final question is, what resources are available for beginners to get started with F1-X?
Yeah, so we offer with each kit a quick start card, which can also be found online. This is a one-pager for kind of helping you get started very quickly at the bench, especially if you're already familiar with Gibson assembly. We have a user guide that's a much more thorough document. We have a fragment splitter tool on our website, as well as an app note on the lycopene biosynthesis pathway.
That's perfect. So that's all the time we have for questions. Again, thank you very much, Jasmine, for your time today and such a great presentation, and also Cosmo for hosting this webinar. We will see you on the next time.
All right, thank you.